All right, so here we are back in class. We have our exam in 1 week from today, and it's our first exam. Here's what you should be doing this week until the exam. So there's the term paper, and you can make an easy start on your term paper by just looking into what the heck's going on. And it's going to be, look at the syllabus and see your term paper is going to be writing about a receptor of your choice, and then the syllabus describes what you need to write about it. Then I made a video on Brightspace about how to actually do that, because it's different. You actually have to start the term paper soon and plug away at it. And the way to think about it is I'm trying to avoid you using very much AI. You can use some, and if you use any AI, you have to include it in your bibliography because it's a source. You know, I've seen AI make mistakes, right? So it's a source. Sources can have mistakes, yeah? But I made these documents so I see the record of your work. And if you just paste your paper in, I'll be like, that's probably AI. And I won't challenge you, because I don't know how to do that, but I won't be impressed. Don't do that. And if you paste in big sections, I'll say, oh, that's probably AI also, right? You can use AI, but you got to write it yourself. And that could only happen by doing research over time, and you have to document that. So a couple of students have already opened the Google Docs. They have. One's for notes, the other's for writing, and they've already said, I think I'm going to write about this receptor. So you should do that, and I know you're focused on your test. Great, right? Like that. Don't worry about it. You don't have to do it every moment, but you do have to do it soon, and I'll remind you of that after our test, because you do have to plug away at it. Otherwise, it doesn't look like you wrote it, and I won't give you a great grade if it doesn't look like you wrote it. You know, I don't need to challenge you and say, I think you're just making this up, you know, and you're just pasting it in from AI. But, you know, it needs to look like you wrote it, which means you're in there typing, because I can see, right, the editing history, because we're a shared document, right? And I'm not trying to scare you or anything, I'm trying to teach you. That's the way you write a paper anyway, is you got to do research on it, you know, and it's not a new or different way, it's just in the age of AI, I need to spread the assignment out and be able to observe you working on it every once in a while along the way. And not just, oh, I'm crunching it in, oh, and I ran out of time, so I'm just going to paste in AI, right? Not the way to go. So that's that. Don't worry about it. You know, it's very much like you're always expected to write papers. We have a week from today. Go over everything. Go over that old exam. Go over all our quizzes. Go everything. Everything's covered. There might be some same questions. I'll hand your third quiz back today. Made an answer key video. Look at that, you know. It'll help you with specific things, even if you've got everything right, because it just helps you tune into what the exam's about, you know. The exam is not going to be exactly like those quizzes. You know, those are really, quizzes were relatively easy because they're very focused on a limited amount of material, and then the answers are really short, direct. The exam's a little bit longer than that. But it's like that same spirit. What's this, what's that, yeah? And it's everything we covered and the chapters in the book that we covered. So it's there. Watch this answer key video, and then, we have one more assignment that's due the day exam. It's about MIC. And there will be a video, and we've done this before. You just watch the video, there's a description of what you got to draw and label, and send me a pic of it, right? And it fits right in. It's part of that receptor tyrosine kinase story. So it fits right in. And people did great on this quiz. I think people have a really good handle on receptor tyrosine kinases. So that MIC video is really important, but it's not a ton of new material. You know, you should have already, you know, read through all the, you know, chapters and the, you know, we're going to have on the exam, stuff like that. And the MIC just should help seal the deal. You have to look at it and learn it, though, because it is new stuff, but it's like the skin cancer patient. We roll into another example of a specific kind of cancer, and you're like, shoot, oh, shoot. Because one way that MIC gets activated is through a chromosomal translocation in a kind of cancer called Burkitt's lymphoma. And that's what happens to those patients, is they just rearrange MIC, and they make way too much of it in blood cells. So you got to watch that video, do the assignment. Don't freak out, you know, that we have another assignment before the exam, you know, because it's working on the exam knowledge. And so that's that, okay? And so then this is where we're at. Oh, there's the, you know, term paper thing. That's just everybody's name that I see. You know, they have 2 of these Google Docs. And everybody should have seen that, I think, gotten an email and said, you shared a Google Doc with you. So look at this. This is question 1 from the quiz. 6 or 7 different students. Which cells in your body have the PDUF gene? All. All cells. All of them. All cells, genomic equivalents. Genomic equivalents. All cells have these genes present, just not active. All cells due to genomic equivalence, but the PDUF gene should only be transcribed in the megakaryocyte in the bone marrow, right? So longer and longer answers, and that's fine, and I'll see that on the exam. Remember that there are 50 questions on the exam, and so if you get bogged down expressing a lot of knowledge in any one question, you'll run out of time, you'll be frustrated. And you won't, because I pointed this out many times. You'll be like, I gotta keep rocking through this and limit it, right? I'm not asking for an explanation here. That's a great answer, all. All cells is that's not even necessary. Which cells? All. Like that. Don't worry about it though. Don't think that you gotta become crisp, because partly sometimes it's good to, share a little bit more, right? But don't feel that you're kind of like sort of showing off or really getting me on my side by saying everything you know about the estrogen receptor. Because remember, we saw that concept map, you know tons about the estrogen receptor. The questions on the exam will be focused, and I won't be saying, tell me everything you know about the estrogen receptor. They'll be about how many domains does the estrogen receptor have, and what are they, right? And if you add more stuff, fine, but I'm not giving you credit for that, right? There's no such thing as extra credit. I should also mention that when I grade exams in all my classes, I go through one question at a time. So I grade anonymously across each question. So I haven't seen what else you've written, So just each question is independent, and I'm looking at it and I'm comparing everybody's first answer on the exam. And I'll just say that because if you ever have an instructor that grades your exam in entirety, that's just a ridiculous way to grade exams. Because there's a carryover where the instructor, I just try to get grading done. If I'm looking at your exams and you're doing, hey, you knew that, you knew that, you knew that, you didn't get that answer very well, but you know it, There's just tremendous bias if you look at one student. Whether you know their name or not, as you roll through their exam, they're either getting a lot right or not. And you just lean into that. You're just like, no, no, no. And so you gotta do one question at a time anonymously. So they stand independently. Any other grading strategy is just a joke, an embarrassment, yeah? Because of that, everybody has that bias. Like, Jesus Christ would have that bias. I'm sorry, that's kind of an extreme example. God would have that bias. Yeah, you should say, when you're being judged by God, you should say, let's not look at my entire life all at once, because you might start feeling like I'm leaning in a certain direction on this person, right? Yeah, no, let's just look one event and then compare it with everybody else's events, like everyone else is a kid, they messed up, like that. So, yeah, sorry. But that's a thing, right? Okay. Another thing about it is there was another question on the quiz, which is, it says, draw and label the parts of the 2 genes involved in the chromosomal translocation. And people did a good job labeling, but just also pay attention, because I ask specific questions. Label, right? So some people didn't label it, right? And I've stopped labeling the genes all the time. I'm just like, here's the gene, right? And I'm talking about the transcription unit and the cis-regulatory region. But when it says label, or explain, or give 3 examples, like that, and it's all boom, 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 like that. Just, don't fret like that's an extra challenge. It's just trying to help you, guide you to studying, and then also, plowing through the exam, okay? Great, super. So Sam told us about CAR T-cell therapy. The next question isn't going to be on the exam. I don't expect this much, and people would call it synthesis. But I have plenty of questions I could ask, but look at this question and the answer I wrote. Just to think about that you should be trying to pull your learning together. So not limited to just a set of flashcards, but really, what is the explanation, right? So look at this, right? Briefly describe the reason for Sam's second talk about CAR T-cell therapy, just Tuesday. Normally, T cells require 3 receptor signaling pathways to be integrated to activate the T cell to kill cells. The CAR gene combines all 3 receptor genes into one gene. The CAR gene is put into cancer patients' T cells, and the CAR protein gets made. Now, just one CAR receptor protein needs to be activated for the T cells to kill target gene. Right? That's it, in summary. Right? That was his point, and that's why he told it that. You know, it's a kind of cancer therapy, and what are they up to? And what they're up to is just exactly that, I think. You know, I mean, I'm not, you know, you could elaborate on it more, right? Like that, talk more about it, but, you know, boom, boom, boom. And it's good for you to try to pull your learning together like that. Now, again, I'm not saying get ready for that kind of question where I'm like, pull it all together. Tell me the big picture perfectly. No, I just wanted to show you that so that you're aware that it's not, we're not just trying to memorize stuff, but we're trying to understand the pieces like that, you know? Like that's great. Yep?
I'm still stuck on the word integration. So when it says the CAR gene combines, that's the integration. At one point, I have in my notes that integration is like checks and balances. Are those different
No,
things?
checks and balances. So back to just the natural T cell. The natural T cell is built to be really careful to avoid attacking the person's own cells. And autoimmune disease is when our own immune system attacks some of our normal cells. And that's a mistake. So the T cell is built to be careful. It has checks and balances. It needs to integrate signals to get activated.
At least 2?
At least 2, that's what Sam said, and it's 3.
Okay.
Yeah. So that's the natural thing, and that's what integration is. It's natural, it's normal, and it is that there could be multiple signal transduction pathways needed to really fully activate a cell response. And those are checks and balances. You know, like, yep, you're good to go. Yep, like that, yep. So yeah, so I don't want to over or underemphasize that. You know, it's good to see that, you know, kind of, it's always good to try to come up with a summary statement of, well, what was that about? Like that goofy student play that I made. I'd never done that before, and you know, I think it was okay. And as soon as class was over, I was like, that could have been a lot better, you know. But it was, you know, like that, right? But you could make it better, right? You could think about it and make it better. I always say, I just have to say really quickly that one time I taught BIO409, and there was a student named Brian. Brian is long gone, so you don't have to worry about Brian. Brian, you never knew. And in class, I talked about transcription regulation in BIO409 for students that were just in intro bio. And I said, the gene is like an airport, and RNA polymerase is like an airplane that's going to land on the runway. The runway is the transcription unit, like that, you know, so I use that analogy. Oh, man, and Brian, you know, just the best person. And Brian said to me once, he said, you know, you talk a lot about airplanes in class, but there's never an airplane question on the exam. And I was just like, oh, yeah, darn it, you know. So, right, that, you know, we do talk in analogies, and you can use them on the exam, but we're trying to understand the real biology underlying the airport or the burglar, you know, analogy, right, like that. Yep, okay. But yeah, questions people have about anything at all? Yep, so we have this big course picture, and we're catching our first exam when we're rolling in it, and we're very deeply into it. And that is, transcription regulation is a big deal on our planet. Signal transduction is a big story on our planet. Cell cycle regulation is a big story on our planet. Those all fit together in cancer biology. Great, we're chasing after that. And, but they're also hugely part of our normal health and disease, not limited to cancer. And then also, you know, all plants and animals and even microbes are all about regulating gene expression. Yep. And they also have a lot of signaling, communication between cells like that. Yep. And then in eukaryotic cells, not prokaryotic cells, they all have orthologs of these very famous cell cycle regulators, like cyclin. There are orthologs in cyclin in every eukaryotic cell on our planet. Yep, remarkably similar. Easy with a blast search to identify tremendous orthologs in the cell cycle between all the plants and animals and the single-celled eukaryotes on our planet. The people who won the Nobel Prize in medicine for unraveling the cell cycle, they studied baker's yeast and brewer's yeast. The stuff that is used to ferment alcohol for liquor, and the yeast species made bread derived. Because they just studied yeast, they never studied humans at all. In fact, of all the Nobel Prizes in medicine that have ever been awarded, almost 90% were for people who did not study people. You know, and it's because of orthologs. Model organisms are all about orthologs, okay? All right, great. So I just wanted to show you this, that in our list of these transcription factors, which of course will be a big part of our exam, the story is the dynamic story of regulation of gene expression. And a good thing to learn is, how is the transcription factor regulated? How is it turned on and off? And we had this one about MYC. MYC is there just, and you know, there's more about that in the video. The story of MYC is, do not make it. Don't make MYC. Don't do it. Like that, right? And so there's a bunch that just get bundled together. So I bundled them together. We have to start a new page almost, because we're going to have some more today. Okay? So we're pretty much done talking about receptor tyrosine kinases for now, with the exception of the MYC assignment that we'll do. After the first exam, for the second exam, there'll be more receptor tyrosine kinase stories, because they come up again and again in our book. So now we're going to go on to these G protein coupled receptors. So it's a different class of receptors, and we're going to have two examples of these signal transduction pathways that we have to learn. So 3 examples up there, and 2 examples down here. So different stories, different receptors, different biology, and so let's just look at that. So these G protein coupled receptors are far more famous than the receptor tyrosine kinases. We really focus on receptor tyrosine kinases because they're often involved in cancer biology, and they're super important. G protein coupled receptors are unbelievably significant in our lives. And we're talking about a class of receptors that all work the same way, basically. So look at this list of stuff that they're in charge of. People will say that about 70% of all pharmaceutical drugs work by regulating G protein coupled receptor pathways. That's how significant they are. I think we have, the number varies somewhat, like that, but you should think we have about 1,000 different G protein coupled receptors in the human genome. And they're called G protein coupled receptors for a reason. They're going to interact with a G protein, a trimeric G protein. Remember RAS story? And that is a monomeric G protein. That's the receptor tyrosine kinase story. These are a G protein. Luckily, they're a very different kind. They're called a trimeric G protein. So that's why they're called G protein coupled receptors. All of the receptors, so there's an example of receptor proteins, all of them, all 1,000 of them in humans and every other organism have a common feature, all of them. And that is the receptor protein passes through the membrane 7 times. So that's just kind of a historical fact. Doesn't have a big deal, but it made it easy for scientists to discover all of them, because they all have that same kind of trademark characteristic. They all go through the membrane 7 times. And so sometimes they're called 7-pass transmembrane receptors. With that, they're all in that same category. Yep.
They just zigzag? Is it like to anchor them more?
Well, so they're transmembrane receptors, so that means they cross the membrane.
Yeah, but like, is that to like send signals through it multiple different ways, or is it to hold it? Like, what is the reasoning for looping so many times?
Yep. And so the reason for looping that many times is like, it's no big deal.
Okay.
In general, you can think of a transmembrane receptor as like a pair of pliers. And when you open and close the pliers, the clamp part of the pliers opens and closes, and the handles open and close And that's the point of the transmembrane receptor, is that this part is really just that, the rivet that holds the two parts of the pliers together that it pivots on. So it does have some structural significance for that. But we have this outside extracellular ligand binding domain that can open and close. And it closes a little bit on the ligand. And when it closes, those intracellular domains, so they have a change in their structure, and that activates them. So the whole point of a transmembrane receptor is it's going to bring a signal across the membrane. The transmembrane region is of not great consequence. The reason that it was easy for scientists to spot them is that they're about, each time the protein crosses the membrane, it's about 20 amino acids, and they're very hydrophobic. So these proteins all have 7 stretches of about 20 amino acids that are very hydrophobic. And you can use a computer to look for that, right? Because there are only a certain number of amino acids that are hydrophobic. And you say, show me all the proteins that have 7 hydrophobic domains like that. Yep. And you do that not by looking at the proteins, but looking at the coding regions in the DNA. So when scientists were able to analyze lots and lots of DNA sequence, they could just search for lots and lots of G protein coupled receptors. Great. So here's how they all function, is they bind a ligand, extracellular ligand, and then they're activated, and they activate a trimeric G protein. It just means it's got 3 domains. Who cares, right? There's lots of details to that, but for our course, it's the same story. They start out, G proteins start out inactive, bound to GDP. Then they exchange GTP for GDP and are active. And when they're active, in this case, it's the alpha subunit. The other subunits fall away, and the alpha subunit is bound to GTP and is active until it breaks the GTP down to GDP. G proteins are called G proteins because they are always bound to either GDP and inactive or GTP and active. And that's the end. And it's just nice that they have for us these 2 extra domain, or 2 extra, not alpha domain subunits, 2 extra subunits that are released when they are active. Because we can see, oh yeah, that's very different than RAS. These are not RAS. And all of the 1,000 or so G protein coupled receptors, they'll work by activating a trimeric G protein, right? And it stays active for a while, so we get some amplification, and then it does a job to bring the signal inside. Yep. All right, and as hugely significant as those are, we're almost done talking with them. I mean, we have to explain now 2 pathways, just 2 of them, right? And so, and there's the book just showing the G protein activity cycle, right? Yeah. And so it's pretty small, but it's in your book, and we can just look at it and see what's going on here. So here is the famous hormone epinephrine. It used to be called adrenaline. The tiger walks in the room, and I'm like, whoa, right? I got to get out of here. So let's just talk about how I get out of here. So a hormone called epinephrine gets released and goes through our blood, and it binds, and this is it binding to muscles. And the muscles have a molecule called glycogen that's a polymer of sugar. And epinephrine is going to cause the muscles to break the polymer down to monomers. And they're going to be monomers of sugar, and that muscle cell is going to have a sugar rush. Literally. And you know what a sugar rush is, right? You eat a candy bar and you're like, I'm ready to go, right? That muscle cell has a sugar rush and is ready to help you get out of there. You know, the tiger comes in and lots of cells in your body activate, right? And are like, oh, that's a tiger. And you are excited really fast. All those cells are getting, lots of those cells are getting a sugar rush. Not from sugar in your blood. They have a polymer of sugar, glycogen, stored in the cell, and they just break it down, and it releases the sugar. And they get all that sugar metabolism, a rush of what? ATP to boogie out of there. Yep. So that's what's happening in this picture. There is no time to activate a transcription factor. That takes a while, you know, a couple of minutes or something, you know. They're like, I got to be ready to rock. And every cell is going to be poised to have a sugar rush like that. And there's some other types of responses, right? Our brain responds partly through a sugar rush, but also through neurotransmitters and stuff like that, and kind of double checking and screaming, you know, and things like that, right? But yeah, so here's the pathway. It's just there's epinephrine binding in some muscle cell. That's what we're thinking it is. G protein coupled receptor and activates GTP, GTP and the G protein, and then it activates this protein called adenylyl cyclase. And that catalyzes the production of cyclic AMP. So I'll just draw this over here, because I always stumble with the pronunciation, because there is this enzyme in the cell membrane, and the enzyme is called adenylyl cyclase. Or it's also called, and I'm used to calling it this, but our book doesn't, adenylate cyclase. And you might have learned it that way too. And what it does is it takes, I don't even remember right now what the substrate is, but it makes something called cyclic AMP, C-A-M-P, cyclic AMP. And cyclic AMP is an example of a second messenger. And we already know 3, they're not involved in this story. It's a different second messenger. So that's nice, because we have receptor tyrosine kinases that can use the IP3, the DAG, and the calcium story. And here's another one. And again, you might have heard second messenger in IntroBio, and when they told you about that, they were trying to introduce you to this reality, but it was you were just getting started in college, right? I wore bedroom slippers my whole first year of college in Minnesota. That was really cool. I was away from home, and I was like, Mom, right? So, maybe you were that out of it too, because I was pretty out of it. But we make cyclic AMP, and what are we doing? We're making it with an enzyme, so we're amplifying the signal, and then it floats into the cell, right? The cyclic AMP floats into the cytoplasm, right? Throughout the cytoplasm, right? So back to this, it's got a target, and the target is called protein kinase A. And you don't need to know the details of this. It's just that cyclic AMP activates protein kinase A. The way it activates it is that it helps it release some inhibitory protein. But cyclic AMP activates protein kinase A. Protein kinase A is going to phosphorylate the enzyme that breaks down glycogen. And so that enzyme is going to be activated. It needs to be phosphorylated to be active, and then it just goes do, 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 do. And we have lots of glycogen, long polymers of glycogen, and each monomer is glucose. So it goes do, 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 and it's just this massive sugar rush. You don't even feel how much of a sugar rush it is because it's in your muscles, but your muscles are jacked to the max. Let's run away or climb a freaking tree as fast as you've ever climbed one, because that's a tiger, we got to go like that, just cranked up. And that's it. That's a good one, a familiar one, right? Unforgettable. Now that's it. It's the muscles responding to the tiger, right? And the rate limiting step is just let's crank up the amount of sugar, and the muscles will be ready to make, you make a lot of ATP, do a lot of muscle contraction, get out of there. Okay? For us, the reason this comes up in our course is because this is an example, and it's only one of a couple of examples that we have in our course where there's no change in gene expression. And so that happens. Cell responses can happen like that, where there's no new gene expression. And people like to call this a non-genomic response. The genome is all the DNA, and we don't need it to make this response. And that's true for many, many responses. There's going to be a response, and you don't need to make new genes. Even if you don't have to do it fast, you just don't need new genes. When Sam comes back to talk CAR T cell therapy the third time, it's going to be about the cellular response. What does a T cell do? And it's going to do several things, and they're going to include an example of a non-genomic response. And what it is this T cell is just going to wrap around the cancer cell. Because when the T cell first bumps into the cancer cell and makes contact, it activates some of the CAR receptors, and its response is just to hug the cancer cell. Because when it does, it makes more contact, and all those receptors activate, and it's like, sorry, but F you, cancer cell, you're out of here. And it's a non-genomic response. It's rearranging the cytoskeleton, and it's just doing like that. And it's just brutal. When you activate a T cell, it's a killer T cell for a reason. It's super rowdy. So we're going to learn about a non-genomic response. There's our favorite. It's a great one, right? The other responses that we're really focused on are called genomic responses. And these cell responses are one or the other or both. The CAR T cell activates both. We'll see changes in gene expression to get the cells to proliferate. We'll see non-genomic responses to get the T cell to hug the cancer cell. Yep, so great, just those two. It's either going to be genomic, non-genomic, or both. This is a non-genomic response. It's unforgettable, right? Good. All right, so on we go. And then it just shows that when we activate the protein kinase A, this is the other one, sorry. We're moving on to the other one, sorry. So that's the first one of two. So let's just scroll back to that to make sure I get that straight too for you. So the first one is called an adrenergic receptor, adrenergic responses. It's epinephrine, the adrenaline receptor. We're focused on muscle cells, the fact that they have lots of glycogen stored, ready to make quick responses. And this pathway does that. G protein coupled receptor pathway activates G proteins, activates adenylyl cyclase, and activates protein kinase A. Protein kinase A phosphorylates that enzyme. We don't need to know the enzyme, just that it cuts glycogen down from the polymer to sugar monomers, and those sugars get burned and make a lot of ATP. Yep?
Are all adrenergic receptors, do they all cause non-genomic responses, or is it just that one example?
No. There are lots of adrenergic responses. Those are, there are tons of different kinds of cellular responses. That's a very specific one. Yep.
Okay.
Yep. Yeah. Yep. Okay. So that's our first example in G protein coupled receptors. We're doing great. So our second example is the dopamine receptor. And there's a really good chance you've heard of that ligand, dopamine. It's a neurotransmitter, and it makes us happy. It's in our pleasure center in our brain. So when you get a runner's high or an illegal drug high, it's the dopamine receptor. It's getting activated through dopamine. And a famous thing that it does is it's a neurotransmitter, and it opens and closes channels in the cell membrane to get the feeling. And so just raise your hand if you happen to have taken neurobiology. So several students have taken neurobiology, and then raise your hand if you learned about dopamine there. It's a super famous neurotransmitter. You probably learned about it some and learned about how it affects neurotransmission. Dopamine also does this. So even if you hadn't had neurobiology, there's a good chance you've heard about this famous neurotransmitter and this, that affects our feelings and our activity levels and stuff like that. So dopamine also does this response that we're going to learn about. And this response is a longer-term response. That first response of affecting how the nerve cells are talking to each other, that's a non-genomic response. Here comes a genomic response, because dopamine also affects long-term changes in our brains, okay? And it's not a neurobiology class, so we're not doing all the responses. Adrenergic responses, too, are probably a big story in Doug Curry's neurobiology course, but this is not that course, and we're not depending on you having taken it, okay? So here's our second example And it looks like this. And again, it's in her book, and I recognize that this is a little bit too far away, and this is going to be an example of a genomic response in a G protein coupled receptor. Okay? And so in this case, it's the ligand is dopamine, and this is a receptor that activates the G protein, and happily for our students learning this, it also activates the adenylyl cyclase, and we synthesize cyclic AMP. And happily for students learning this stuff, the cyclic AMP in this example activates protein kinase A. But in this example, instead of phosphorylating the enzyme that breaks down glycogen, it phosphorylates a transcription factor that's called CREB. And CREB activates transcription. So I'll just show you the reason we're talking about this is CREB fits in here. Phosphorylated by a kinase. Our examples are, well, FOXO gets turned off, ALK gets turned on, CREB gets turned on. We have to have a few of those because lots of transcription factors get turned on or off by phosphorylation. And CREB is a really good choice. And I'll just put over here. Oh, oh, no, I won't go to that right now. I'll just put over here so I write it down so you can see. Because what is CREB? What is that, what's that, how's that spelled? Actually, I should just leave this here because we're going to activate adenylyl cyclase. The cyclic AMP is going to, in turn, activate protein kinase A. And then the protein kinase A is going to phosphorylate CREB. And CREB is spelled like this. And it's a transcription factor that needs to be phosphorylated to be active. Right? And the test could just be like, what's a signaling molecule that activates CREB? Dopamine. What does CREB do when it gets activated? It's phosphorylated, it's a transcription factor, it activates target genes. So again, we just learned this term today, but all these examples are examples of genomic responses. We're going to have some new gene expression. And it's spelled CREB, and you'll see that's a memorable little acronym, because now we'll just draw this. So here's a cell, here's dopamine, and there's a gene. And again, I don't label these parts, but you probably will want to do that on the exam. And so years ago, the scientists were like, how does dopamine work? Biochemists, they're like, how does dopamine work? And so they just took cells and they either added dopamine or not, and then they ground them up. And they said, what are some of the molecules that are different in them? And so they found that one of the molecules that's different is when dopamine's around, we get cyclic AMP. Yeah? So they found that pathway to activate it, and they're like, oh, okay, so dopamine's working through a cyclic AMP-dependent pathway in this example. Not in every example. Dopamine can also act through non-genomic pathways. I mentioned that. But in this example, which is really a significant example, it works this way. And then they're like, I wonder what it does. So then some molecular biologists were checking into it, and when dopamine's around, they saw new genes expressed. Like that, right? So just the experiments historically. And so they said, it's not dopamine that's activating those genes. It's cyclic AMP probably, or something on that pathway inside the cell. So they called these genes cyclic AMP responsive genes, or response genes. They showed through experiments you need to have some new genes expressed for cyclic AMP to have an effect. What are those genes? So they found some of those genes. And so what did they do then? They did those deletion studies to see what it took for this gene to be responsive. They mapped the enhancer by doing the footprinting experiments and the gel mobility shift experiments. They sequenced the enhancer. They got the enhancer. They said, we got to go after this protein. What is this transcription factor that's bound there, right? And in the process, somebody's like, what should we be calling these enhancers? And they said, well, it makes sense to call those enhancers cyclic AMP response elements. And we've heard that term before. For some people like to use that for enhancer. Great. Yeah. And so they purified this protein. They used an affinity column, and the people were working hard purifying that protein. And they said, well, they were biochemists. And biochemists go like this for naming. They're like, well, let's call this cyclic AMP response element binding protein. Right? They're not making, any more stronger claim than just what they're really studying. Cyclic AMP response element binding protein. And what we'll do is we'll just call it CREB for short. CREB, cyclic AMP response element binding protein. So it's a cute name and a satisfying name, because that's how biochemists often work, is they're like, let's make it make sense. It's geneticists that would call this something like armadillo, and you'd be like, come on. And you're like, they're cute, they are cute, armadillos curl up, they're cute. And you're like, yeah, okay, good for you, right? So that's that one, and that's our example there, is dopamine activates this pathway, and it's a genomic response, and it just adds another one of those transcription factors to our story. Here's the structure for cyclic AMP. Am I going to ask anything about that on the exam? No. Do you want to go there because you know a lot of chemistry? Yeah. Right? But no, we don't need to know the structure of cyclic AMP. We're probably, if we ever need to know the structure of a molecule, we'll say, you need to know the structure of that molecule. Yep. Yeah, so just a little navigation there in your book, and ask questions about that. And so there it is, cranking it up. Yep, and that's that pathway, right? And there's CREB, and we're good. And and so we're all set with G protein coupled receptors. I just want to show you just 2 more examples, just to show them. And in one of the examples, we'll learn a term, okay? So, and they have to do with smell and then vision, right? So, in our nose, I should just show you in this first. Whenever we smell anything, it's because a molecule of smell, like cinnamon, molecule actually binds to receptors in our nose, literally. That's the only way we ever smell anything. And so in this case, with our sense of smell, it's all about the G protein coupled receptor. You don't need to dive into these terms. There's a different term, and I'll show you it. The molecules are called odorants, and the receptors are called odorant receptors. And you probably know that most mammals, like the dog Maisie, can smell way better than we can, way better sense of smell than humans. We don't have a very good sense of smell. Mammals evolved initially from some nocturnal mammals that were had to get through things and find things in the dark. So they really emphasized their sense of smell. And so there were lots of gene duplications in the odorant receptor genes. Hundreds of them, so that the mammal could smell with great sensitivity lots of different odorants. And in general, mammals can smell really well. Our primate ancestors then became diurnal, active in the day, and they started to put less emphasis on smell and more on vision. And when that happens, if you don't use it, you lose it. So in the human genome, we have hundreds and hundreds of odorant genes, like our mammalian relatives. And most of ours are broken. They're there because they were there and active, functional historically, but they've broken, and it just takes a long time for them to completely disappear. The initial mutations make them defective, knockouts like that, and we're just losing them. We lost a bunch, right? And so we just want to learn this term. Uh-oh, where'd it go? And that is that it says there are about, 636. That number's not a big deal. I said like hundreds of them. That's great. Hundreds of odor receptors in humans. And only, so 636 odor genes, only 339. Again, not an important number, just, less than half of them or half of them are function. But you can see the other ones, they're there, but they're all broken. And when genes are broken like that, there's a general term, and the general term is they are called pseudogenes. And so that's a wonderful example for you to never forget that term. Pseudogenes are one of the most fantastic examples of the history of evolution on Earth. We have tons of pseudogenes in us. About half our genes are pseudogenes. And they're just disappearing, and it just takes a while for them just to disappear. Smell is the perfect example. We just aren't emphasizing smell. We look around. We are active in the day. Perfect example, to never forget that. Half our smell genes, and in a dog, rat, mouse, cat, theirs are active. They can smell, it would be wonderful to imagine what that must be like. With like a dog with its head out the window, and what they're smelling is must be crazy. The detail and the variety, because they have all those odor receptors and they're all working, because their species of mammal continue to emphasize the sense of smell, right? They can see pretty good. You probably know dogs can't see as well as we can. Yeah. We actually, when we started losing the sense of smell, actually gained by gene duplication another color vision receptor in these old world primates. Yep, okay, awesome. So that's that. Then I just want to show you that this vision one, and maybe I don't even have a slide for it, but where did it go? Yeah, so just another one, not important for our course, not in our book anymore, but our vision all the time we're seeing stuff, it's a G protein coupled receptor activating cells in our eyes. So just how significant our G protein coupled receptors, just monumentally significant. So again, we're taking it easy on that because we put a lot of energy into receptor tyrosine kinases. And they're fascinating, and we're learning the principles of genomic and non-genomic stuff like that, okay? Great, any questions about that? I think we're doing great. I think we'll probably skip concept chat today just because, we'd like to get through as much of this material to be on the exam, so there's not very much new material on Tuesday. You wouldn't want any new material, right? And we're really close to that because the rest of chapter 16 are just some isolated examples of other types of receptors. So these are not receptor tyrosine kinases, of which there are about 60 in humans, and they're not G protein coupled receptors, of which there are, at least 1,000 in humans. These are just different ones. The reason they're there is because there are lots of other types of receptors, and some of them are super, super famous. And super famous not just for humans, but some of them are found in every single animal in our planet and just play gigantic roles. So they kind of have to be in our book because you would be like, I know about hemoglobin, and some of these receptors make hemoglobin just look like a waste of time because these are found in every animal, and play, they play a role in the embryonic development of that animal and every animal, and there would be no animals without these. So our book has to put them in. For us, they're great because in a real 2-point question on the exam, we can roll through them because they are all focused on genomic responses. So what about these receptor non-tyrosine kinases? So what's his name, Sam, that other student, that Sam, he mentioned it. One of those is going to get activated in that pathway. Who cares? Us, there's one of them that activates this thing that people just call JAK-STAT. JAK is the receptor, and STAT is the transcription factor. Easy as pie. And our book shows it. It looks like that. And you just glance at that and just go like that. It's just, Sam actually showed that from her book. You can look at that a little bit to understand it. But again, we're focused on a 2-point question. Give me an example of a non-receptor tyrosine kinase, and you're like, JAK, STAT. Explain it. JAK's the receptor. STAT's the transcription factor. How does STAT get activated? It's phosphorylated. So STAT goes there. And there are lots of non-receptor tyrosine kinases, but JAK is just a great one. And JAK STAT is a great example. Yep. So we go like that in our thing, right? We had to make some space here because we're adding some more. Okay, here's the next one. Okay, so the next one is called, the receptor is called frizzled, and it's the receptor for the ligand Wnt. Yeah, and it's, who do you think named this? Do you think biochemists came up with the name frizzled? Nope. No, geneticists, fruit fly geneticists are like, that mutant fruit fly looks kind of frizzled, kind of put upon, kind of frustrated little mutant fruit fly, super dead, because it can't live without it, So a dead, frizzled embryo. They're like, oh, yeah, let's go to lunch. So another unique type of receptor, super, super important. Orthologs found it everywhere, not just the Wnt and the frizzled, but the whole pathway. So what happens there? And our book shows this, and what shows this is the pathway. What happens when frizzled is activated by the signal pathway Wnt is that a transcription factor called beta-catenin gets stabilized. And when it gets to do its job, it activates transcription. So in this example, what's happening is that we stabilize the protein. And we said that happens with MYC too. It's not a very stable protein. This is kind of an extreme example of that. That's the rate limiting step for Wnt signaling, is we have a protein in there that is made all the time and just gets destroyed as fast as it gets made. And the protein is a transcription factor called beta-catenin. Yep. Let's just look at this figure. I'll just show you this because we'll add a term here because, you can see we can just flow through this, pretty quick. So let's just show this. I'm going to draw this little part here, and you'll see, right, there are two situations. The first is the cell with no Wnt, and the beta-catenin is getting destroyed. It's just destroyed. And then when Wnt shows up, the beta-catenin gets stabilized and gets to do its job. But you see there's another protein here. And so I'm going to just draw that out because we'll learn another term that's generally significant that we just haven't run into yet. So here's a cell. And this cell could be in a jellyfish, which is just about the most distantly related animal to us as we could find. And it has this entire signaling pathway, ortholog of Wnt, ortholog of frizzled, ortholog of beta-catenin, and everything else in the pathway. Super ancient tool for putting together a multicellular animal that was used so long ago it's found in every animal on our planet, but not in any plants, because the last common ancestor between humans and plants was a single-celled eukaryote, and it hadn't figured out how to make a multicellular organism yet. And the species that came down the pathway of making animals very early evolved this pathway because it's used in every animal. And it's not used in plants because they evolved other plants. So it's just a crazy, like, if you want to talk about like the history of evolution on our planet, and like this is like a children's bedtime story. Like I told my kids this bedtime story all the time. They're like, tell us about Wnt, Dad. Because it's so awesome, right? Because I just punch them. I just hit them. Yeah, this is World Wide Web stuff. Yeah, they're growing up. They're older than you, so you can't even call social services. But I'd be like, yeah, I would get like the couch pillows and I'd hold them like cymbals and I'd just crank on them. And I'd be like, there's no Wnt in animals. I mean, plants, and I'd just hit them. Yeah, with the big couch pillow, right, like that. And they wouldn't, they loved it. All right, so this is how Wnt works. So there's Wnt, and it's going to bind to its receptor frizzle.
You should draw it frizzly.
Huh?
You should draw it frizzly.
I should draw it frizzling,
Receptor.
get some frizzling action going on there. Yeah, it's frizzling. And when there's no wind, this beta-catenin gets destroyed. And then, when the wind binds, beta-catenin gets stabilized. And so there's beta-catenin, and it's a transcription factor. But look what's going on with the target gene. So there's a target gene, and it was over here too, you know, in the absence. And all the time, there's actually a protein bound to the enhancer. And that protein is called TCF, and it's a repressor. So it's actively turning off these target genes. It's like you go over to turn on the light switch, and I'm standing there going, yeah, good luck, Alex. Right? TCF is just holding the switch down. So when Wnt gets stabilized, Wnt actually binds not to the DNA. There's no DNA binding domain in beta-catenin. Oops. So TCF is there trying to turn it off. But this active beta-catenin just binds right on it. So there's no DNA binding domain on beta-catenin because it binds on that. So beta-catenin is a transcription factor with no DNA binding domain. It relies on another protein, TCF, that has a DNA binding domain. And what it does is beta-catenin blocks TCF and switches it. And the term for these type of transcription factors is they're just given a special name, and they're called co-regulators. So a co-regulator is a transcription factor that does not have a DNA binding domain. And they're either going to be a co-activator, and that's what beta-catenin is, or a co-repressor. And beta-catenin is a co-activator. And that's a great term, just to kind of expand our understanding of transcription factors, is that most of them have DNA binding domains that bind to the enhancer and get the job done. But some don't. They don't have a DNA binding domain. The target genes dependent on another transcription factor. This is a good example. Have we learned about other examples of that? Yes. Estrogen target genes are regulated by co-regulators. One is a co-repressor, histone deacetylase enzyme. The other is a co-activator, histone acetylase enzyme. And I don't think we used the terms then, but those enzymes are considered transcription factors, and they're called co-regulators. And they could either be a co-activator and acetylate the histones and open up the chromatin, making it accessible for gene expression, or it could be a deacetylase, and they could deacetylate it and close the chromatin up. Yeah, you Sarah?
So it's a co-activator because it's activating transcription, but
Yep.
it's repressing TCF.
Yeah, that's a good point. And that gets to be kind of frustrating, right? When you say we have the TCF repressor and beta-catenin is repressing the repressor. Yep, yeah. And it's doing that. It also has an activation activity. So it's repressing the thing that the TCF was doing, but it's also bringing an activation. So it's a, it's like again, it's like this, where you're like, is this light switch staying off? Yeah, because it's built that way. It's going to stay off. And I'm like, yeah, we're just making sure it's off. And then Alex comes up. Alex's beta-catenin, stabilized beta-catenin, has the strength to push up and get it going. So not just relieve the repression, but actually push it up and make it happen. So molecular biologists love that because it's a really beautiful molecular switch. We would like these genes to be off, fully off, or on, fully on. So let's have a repressor, keep them shut down, and that's what the estrogen receptor is like too. It has a different way of getting it done a little bit, different story, but the genes are really off because it's a repressor, and then they're really on. Yep, so that's a good one, and I think we only have really one official one left. And so we talked about its stability, And then the next one in here, also super famous. Here's an example of how famous this one is. It's called, people call it notch. The notch is the receptor in this case. Usually people will name things like dopamine and Wnt and so forth, and they're referring to the signaling molecule. In this case, they just call it notch, and it's the actual receptor. No big deal. But you'll see there's a reason why, and it's because notch is a super funky, cool protein. Here's an example of how cool notch is. So lots of students do this research internship down at Maine Health Institute for Research. In this class, people have done it. I don't remember who right now, but maybe no one has. Maybe no one's down there. Down there. Yeah, if you're going to be at USM next year and you like these topics like this, that's what all the scientists do. And I'm in charge of it, and the applications are going to go out in about a couple weeks. And you get paid $8,740 during the school year to be down there. Oh, sorry, Joseph was down there. You can talk to Joseph about it. Yep. So it's a state-of-the-art place and stuff like that. And so one time we were down there, a bunch of students and I were down there touring it, and one of the scientists said, everyone in this building studies notch. Because it was a big deal then. Now scientists have learned a lot about it, and they're still working on it. But there was a time when, people had notch T-shirts and stuff like that. Down in the old port, they're like, come on, get one of these notch T-shirts. Maybe it wasn't that popular. Maybe it was. So here's notch, and it's also a memorable thing, and it's just a surprise for us. So when notch gets activated, so here is its ligand. It only has a couple of ligands. The most common one is called delta, but it's the receptor notch that's the curious thing for us. Because when it gets activated, and a protease actually cuts it, and part of notch becomes a transcription factor. It's called the intracellular domain. We don't get any amplification. Think about that, right? No amplification in this pathway. 1-to-1. You activate a receptor, you get a transcription factor. The reason that it works that way is because this delta doesn't float around. The delta is on the surface of other cells that are touching this cell. And so we can just activate all the notch, and we get all the transcription factor going inside. So it's one other example, and our book has to show it because it's so famous. The receptor gets clipped by a protease. Proteases are enzymes that clip a protein. And we release a transcription factor, Q. And I could ask you to draw that, explain that on the test. 2 points. 2 points, 2 points, 2 points. We're doing great. I think somebody had a question, maybe? Okay. And because we're so close, our third one is just another receptor. It's called TGF-beta receptor. They're part of a giant superfamily of receptors. And they work by phosphorylating a target transcription factor called SMAD, and the SMAD family. So we just get another genomic example out of the deal. Our book shows this. There it is. And there's a bunch of different ligands because it's a super family. But it's not really dealt with in our book very much because it just has such a kind of boring activation pathway. It just phosphorylates SMAD, and they go into the nucleus and activate transcription. Yay. So that's the list from our initial handout. Oh, maybe we didn't get on there. I should have made another one. Oh, no, it's up here. SMAD just go up here. Phosphorylated, activated. So you got to look at those figures, but that's the last maybe third of our textbook. The textbook has a couple more of those. Sam actually mentioned one on Tuesday, but those just they're not even as important as this. And these are important because they add more stories to our understanding of transcription regulation, but they're not, what did we dive into? Receptor tyrosine kinases. We still have to learn about MYC and its role in the receptor tyrosine pathway. There's just tons to learn more about that. We learned a little bit about G protein coupled receptors, They're super famous. These are just introductory things, right? Like that, okay? Because we are solid, and we're going to keep talking because that's the material we need to cover for the exam, plus the MIC assignment. So on Tuesday, we'll roll forward with new material, but we can do things like talk through all this stuff, right? You know? And so it would be foolish not to have a bunch of questions in class on Tuesday. Because if there aren't, or whenever you're done with questions, just roll on to new material that's not on the exam, because we don't have time to waste, right? But, you know, so Tuesday's only a review until you're done asking questions. And then we'll have our exam a week from today, like that. And I think, you know, the time's pretty good, right? Because you'll see the MIC assignment is new stuff, really important stuff, but it just fits in, and it'll be nodding, and it'll be like, ooh, wow, like that, okay? So now, anybody have any questions about anything at all? Yeah.
Can you go back a slide to all the receptors?
How about this? No, Matt. Do you have like $10? Wait, has that ever happened to you guys, where somebody has to boost their tuition? You guys already pay some elevated tuition as STEM students. Did you know that? That USM no longer has even tuition across the different majors? Who knew that? Raise your hand if you knew that. Juliet, Matt, Justin, Brianna? Yeah, you pay a little bit more now. It's happening, right? You pay a little bit more
Do
just
STEM
because.
professors make a little bit more?
Huh?
Do the STEM professors
Thank you
make
for
more?
writing that message to the president of the university. Do the STEM professors make more because I'm paid more? No. No, I can't even buy tacos. I just had to beg. Anybody got a taco? Yeah, yeah. So yeah, no. So yeah, do you want to go up a level in tuition, Matt? Because I can go back a slide if you have cash. That one.
No, one more.
Oh, $20, man! Come on.
So for the exam, we need to know all these receptors and kind of be able to draw them out. And if the book gives like extra examples of, say, like a nuclear receptor, we don't really need to focus too much
Yeah,
on that.
the emphasis is the heavy emphasis on the exam is the stuff we covered in class. The reason you have to look at your book is first and foremost to learn really well the stuff we covered in class. So we talked about the estrogen receptor in class, and the most likely questions on the exam that would be from the book and not from that weren't covered in class would be some important feature of the estrogen receptor that you might be like, wow, that's a pretty important feature of the estrogen receptor, like that. But then you spread out further and further. Yeah, it's this hierarchy or pyramid of learning where our whole point of the class is to try to frame the most important stuff, and then you spread out into the book. You would be a mistake to say read page 1, 2, 3, 4, because they're not equally important. Instead, you have to do active learning where you're looking in your book, kind of desperately trying to find the most important things, and the most important things are the stuff we talked about. But there'll be some points on the exam. But that's the later part of your studying effort. Yep? Yes.
So what I've been doing for studying is kind of organizing things into stories, like you said. Like, so in that case, like these are our stories, and like they have different like chapters and stuff like that. But like, would you say that like these are our stories?
Well, they're ones we talked about in class.
Yeah.
But then, you know, we turn to them for different reasons. Like, for example, the pseudogene story, that's broadly significant. And we just tried to grab one example.
Mm-hmm.
From our class today, we learned the term non-genomic response, right? That's super, super common, probably more common than a genomic response, you know?
Yeah.
But we're hanging we make these frames and then we hang our little knowledge facts on the different frames.
Mm-hmm.
And so it'd be nice to have a whole bunch of different drawings, and I say make them big and then decorate the drawings with flashcards, because there's a lot of flashcard knowledge in here, right? Like SMAD, and you turn it over and it's like a transcription factor activated by the, TGF-beta receptor pathway. And that's it. That's our total knowledge. You don't even have to have a drawing for SMADs. Yeah. Back to Matt's question, yeah, I'm not going to ask you to draw the TGF receptor signaling pathway. So you don't need to memorize that in more detail. Yep. Yes.
That was my question, was if we need to be able to draw all of those, or just the MAP kinase?
Well,
And,
certainly the stuff we talked about, and then it quickly falls down to less significant stuff. And understanding it, so you can draw it and label it, or describe it. Yep. Yeah, and all these, right, you've seen, we didn't spend very much time in class about these. These are not going to rank really high on our exam. But we talked about them some, so get ready for those, yeah? Yeah, yeah, and I always tell students, if it doesn't work out on that way, my car is like a red Corolla, and just go slash the tires. right? You know?
First
I
of
mean,
all-
I did that most, I don't know, not half the classes in college. The professor's like, oh, dang it. And there was no Uber then. They're just like, shoot, I gotta walk home. Oops. I used to tell students to steal my car because it was a really junky Honda Civic. And I'd say, I just left my keys in the car. And if you don't like the exam, just take it. Yes, Alex?
First of all, I have a red Corolla also, so don't.
Oh, see, there you go. There you go. Don't hurt your classmates, because that's the wrong message. Yeah, don't slash your classmates' tires and get confused about whose Corolla is that. And so take it easy.
Can
Yeah?
you go forward 2 slides again?
Oh man, I'm making so much extra tips today. 2?
Yeah.
Oh, yep. Yeah, oh, don't forget, I'm trying to record this. Yes. So hopefully that worked. It has so far. Looks like it